So the second method of inhibition is competitive inhibition. What is competitive inhibition? It is something like a lock. You know, that a thief is supposed to carry a master key. So similar type of thing. There are some enzymes which have their active site, which active site is always matching with the substrate. Sometime there will be a competitor which will go and attach to the same place because its shape is matching with the active site. So for example, succinate dehydrogenase or succinate dehydrogenase is the enzyme. So it causes the dehydrogenation of succinic acid. So it joins, what joins to this active site is the substrate called succinic acid. And if it joins, this reaction will cause succinic acid to change into fumaric acid. But there is a competitor molecule which have similar type of shape to this shape, which is called malonic acid. So it may compete with the substrate for the active site. So the active site is always competed for. The competition is between the substrate and the competitor molecule. So this type of inhibition is called as competitive inhibition. Remember, if the concentration of substrate increase, this inhibition would be gone. That's why this inhibition is called as temporary competitive inhibition. Now, there can be non-competitive inhibition also. Now, what exactly is that? We have already learned the allostric thing, remember? So there are some compounds which join with some other place, not with the active place. So non-competitive inhibition can be reversible or irreversible. Reversible one is the one, like for example, the glucose joins with ATP to form glucose 6-phosphate and ADP. Now if there is excess of glucose 6-phosphate, then this will join with the hexokinase enzyme, join with the allostric site. That's why this inhibition is allostric inhibition or retroinhibition, and this will stop the enzyme's activity. As long as glucose 6-phosphate is in the excess amount, this reaction will stop. So this can be called as, remember, inhibitory, or you can say repressible type of enzyme. Always functioning, but will stop functional when there is excess of this. Second category is irreversible non-competitive inhibition. For example, cyanides joins with the copper ion of the cytochrome oxidase enzyme. This enzyme is involved in respiratory system. So once the cyanide joins to the active site has copper ions, then this enzyme will become permanently non-functional and death of a person will take place.